Dr. Maher, Dr. Spira, Chunyu, it's so great to see you. Thanks for joining us on the program this morning.
Thanks for having us.
And I'm real excited to talk about your study and the relationship between activity and rest and rhythm. We'll get into that in a minute. But Dr. Maher, let me start with you. I'm going to ask a softball question here. Can we slow? Have we figured out a way, we meaning the medical community, figured out a way to slow biological aging?
Again, thanks for having us. Among the things we know that there are multiple markers, including the ones we used in the study we're about to discuss. This example is methylation of specific changes in biology that happen over time as we age. And as you would expect, many studies have looked at correlational relationships between particular lifestyle factors, socioeconomic factors, medication usage, and other environmental exposures, and correlated those with differences in markers for biological aging. And these markers for biological aging are derived because they predict things like all-cause mortality or something that is called health span. And so among the things that we know impact the lifespan and health span, those tend to be correlated with decreased biological aging. And so in theory, if you at any age change your exposures or your dietary patterns or your lifestyle, started exercising, quit smoking, quit heavy drinking, things like that, we would expect that you would see over time a decrease in biological age.
So, Dr. Spira, with that in mind, thank you for that, Dr. Maher. Let's talk about the study. I think the study, and I'll let you kind of paraphrase it, but it really focused on activity rhythms and resting rhythms. First, let me ask you, as a layperson, I'm not a practitioner, I'm not a biotech gentleman person. When we're talking about rhythms, what are we talking about here?
Thank you so much. Yeah, that's a great question. And I come to this research question really as a sleep researcher. A lot of my work focuses on sleep. And one of the and actually, we have many different rhythms that our physiology sort of oscillates by on a roughly 24-hour basis. And so you've probably heard of the term circadian rhythms.
Yes.
Right. And so many different aspects of our physiology follow a circadian rhythm, meaning they oscillate or fluctuate roughly once every 24 hours. That comes from circa, meaning approximately, and diem being day. So we've got circadian. And an example that is probably the most salient to most folks of a circadian rhythm is the alternation of wake and sleep. But many different, physiological processes like gene expression and hormone production and core body temperature also follow a circadian rhythm. And what we were talking about in this study is, I'm going a bit deep here, so
That's
get me
okay.
back on course if I'm going too
You're great.
deep.
You're doing good.
But the circadian rhythms they're guided by the master clock in our brain, which is in the front of the hypothalamus, and it's called the suprachiasmatic nucleus, or SCN for short. And that part of our brain coordinates clocks throughout our bodies, in all of our cells, that oscillate according to the instructions of the master clock, ideally, roughly 24 hours, roughly once every 24 hours, following this, this circadian pattern, even in the absence of any stimuli from the environment, like light, etc. That is the definition of a circadian rhythm, as something that follows a diurnal pattern that is under the control of this clock, and it happens endogenously without any input from the environment. What we did in this study was take a relatively distal measure of circadian function by looking at rest patterns of activity and rest measured at the wrist using accelerometers, right? We all are familiar these days with wearable devices. They're ubiquitous. We use research grade devices in this study to look at, to capture basically moment to moment changes in activity over a few 24-hour periods. And what we're able to do with that is come up with basically curves on over a daily basis that we can sort of, average across people, within people, and then compare people in terms of those curves. And we can get various measures from those data, including measures of rhythm strength or fragmentation, etc. And what we did in this study was look at the extent to which those different metrics that we can derive from those moment-to-moment measurements of activity versus rest. We looked at differences between people in those different parameters in relation to their biological age as measured by these epigenetic clocks that Brion was referring to.
So, Chunyu, let's talk about the participants in the study, because you obviously need I would imagine you need some level of human participation in order to do these measurements. Can you tell us talk about the profile of those that participated in the study?
Yeah, so like we use the Baltimore Epidemiologic Catchment Area study. So like it is very long cohort from like 1980s to now, and we use a wave 5 participants, which is like recruited around like 2018, and most of them are in their middle and older age. So the average age is around like 68, and we have around 50% males and females, and we have around like 80% white people, but we still have 20% like Black people, and there are still like a little bit number of the Hispanic and the native like the Americans. So yeah.
So Dr. Spira, I mean, obviously it's a good pool. Why 68-year-old, the average age of 68-year-old? Wouldn't younger people have an impact on the study? Wouldn't that be maybe not predictive, but provide some data that would help maybe formulate maybe treatments in the future, additional research?
That's a great question. And the thing is, we have been following this sample since 1981.
Okay.
These are people who have been part of a study, a very important study actually, that provided some of the first evidence for the prevalence and one-year incidence of mental disorders in the United States. So, and this dates back to Jimmy Carter and his commission on mental health. And that's a whole other interview.
Yeah, but it just goes to show you that you're looking, I guess you're tracking these people over this long period of time. You need to do that. It's not like you can go back in time. You have to, in order to test aging, you have to look at people as they age.
Exactly. And Brion and I and our colleague Bill Eaton and some other faculty at Johns Hopkins and at the National Institute on Aging have been working with this population or with this cohort. Now that it is primarily older adults, right? We had some people who were in their 50s, but I believe now they're all older than that, at least 60. And, we've been studying them, and this was an opportunity. This is one of the many, many research questions that we can ask of this aging cohort. And we thought it was a great opportunity to ask this question because we had the monitoring data from the wrist accelerometry or actigraphy. And we have this wonderful epigenetic aging data. So that's why.
So let me, and I apologize because I only have a certain amount of time, but clearly we're going to, this is an ongoing amount of research that we're going to have to continue to check the pulse of, pun and fully intended. But Dr. Maher, let me come to you, because we started with, can you slow biological aging? How do you take what you've learned from this study and can you apply it practically? Like, can Jeff Snyder take this information, go to his primary care physician and then say, Jeff, you need to do this, 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 and you're going to slow your butt. You're not going to be 54 anymore. You're going to be 44. I mean, what do we do with this data set and this research that you and the team have worked on together? What's the next step?
Well, I think it's a very good question, and I think over the next decade or so, we'll understand more about this process, and if there are targeted approaches to improving, say, in a personalized way, a person's epigenome. But as I mentioned, the general things we currently understand as improving health will have an impact on genome-wide methylation and also site-specific methylation in regions that are relevant to health. And so my recommendation to anyone is that exercise, eating well, maintaining a healthy BMI, those are the things that are most, and obviously maintaining a regular sleep pattern, which is what which is what this work shows, are things that are really important. And also, generally attempting to avoid stress or limit your responses to stress. Those are the kinds of things that there's pretty compelling evidence impact your epigenome. And like I said, over time, I think we'll understand better if there are ways that you can target particular regions. I think one thing that's important to note is one of the reasons we do this kind of research is not just to say, hey, there's this general impact on aging, or there's this general in this direction or that direction, is also for us to understand, because these aren't anonymous regions in the genome that we're measuring. These are in genes. And so we can understand the specific biological pathways that are impacted related to sleep or impacted to stress, related to stress exposures.
Well, it's absolutely fascinating. As a layperson, those are things, I think you gave us good takeaways. There are things we can control. There are a lot of things out of our control. We can't control the sun. We can't control our climate to a certain extent, but we can control certain things. We need to take care of ourselves. And obviously the research will continue. Dr. Maher, Dr. Spira, Chunyu, great to see you. Thanks for joining us. Great research. And look, we look forward to having you back on the program again very soon.
Thanks, Jeff. Thanks for having us.
Well, thanks for watching. And don't forget, we're back again tomorrow morning with another great expert and another great topic. You can tune in right here Monday through Sunday at 7:30 a.m. Eastern Time. And don't forget to subscribe to our daily newsletter, The Morning Pulse, for all the news in one place, delivered directly to your email box. Check it out at our website.